Okay, so I think my slide is up.
Yes, we can see the slides.
Okay, thank you. So today, the topics that we will be going through will be rehashed or slightly refresh a bit on the nanopore sequencing and go deeper into the applications of tumor normal sequencing workflow, and then how do we analyze the data that have been generated by using the tumor normal sequencing workflows. So introduction to nanopore sequencing. So this one actually we have kind of introduced to you every webinars, but just to quickly rehash again. So ONT is a UK-based company who develops and sells the nanopore sequencing products, including the DNA sequencer like the MinION, or some people prefer to call it the Minion, and then another types of sequencing machine, ProMin-Ion, which is a larger scales of the nanopore sequencing device. And then the sequencing technologies will be able to sequence the DNA molecules at the single molecule level. And then it's a real-time sequencing, so we can get the data while the sequencing is still ongoing. So these technologies have been used in many different research domains, and then actually have been adopted across more than 135 different countries. So the publications are at the moment more than 14,000 or 15,000, and then the number is still increasing. So the nanopore or ONT, basically their technologies is used in many different by many different community members, and then the device have been tested in many different environments as well. They have bring the device to the field and then all the way to the space to try out the technologies, and then the sequencing have been applied in many different industry as well, for pathogen detections, to sequence the microbiomes, and then of course to sequence the human genome. So what are the benefits of using the nanopore sequencing compared to other sequencing technologies? First, of course, it provides richer insights. So because it's measuring the sequence or the DNA molecules at single molecule level, it will be able to detect directly the native DNA or RNA. So if let's say the DNA molecules are kind of methylated, and then we are also able to capture the epigenetic data from the same sequencing run. So perhaps in the normal whole genome sequencing of human samples, we might need to use multiple libraries or multiple applications like whole genome sequencing and then the bisulfite sequencing in order to capture the genomic context or genomic information and also the epigenomics information. But using the nanopore, we are able to capture multi-omics information in one sequencing run. And then another benefit will be faster time to results. So the data generated is real time while the sequencing is still ongoing. So we can monitor very closely on our sequencing outcome. So if let's say the sequencing is running fine, we can let the sequencing be active throughout the 72 hours per sequencing run. And then if let's say for some simple cases like sequencing bacterial genomes, when we observe sufficient data have been obtained from the sequencing run, we can actually decide whether to stop the sequencing and then wash the flow cells and then keep the flow cell for the next use. So this one is important for some urgent cases, like we need to know the answer very quickly from the sequencing. And then of course, the sequencing devices at the moment is kind of compared to other technologies, they are the most accessible and most affordable. So the size of the sequencing machine is small, and then that's why they can be brought to many different locations or relocate very easily, and then do the sequencing very easily from different environments. So the outcome from the sequencing run, like the data formats, are all the popular one. So you can use many different software or analysis pipelines to do the data analysis once the data is obtained. So how the nanopore sequencing works? Of course, the core of the sequencing technologies is the nanopore. So this is the heart of the technology. So this nanopore is actually a protein. So it has a nanoscale hole inside the proteins, and then it looks like this. So this actually, the natural functions of this nanopore is actually connecting two different systems or two regions separated by a membrane. So this nanopore protein normally is located on the membranes, and then it lets ions or any molecules pass through the membranes through the nanoscale pores inside the proteins. So for kind of, I think it's easier to understand how it works by looking at the animations here. So this nanopore is fixed on the membrane, supporting membrane. And then when the DNA or RNA molecules are brought over to the pores through the motor enzymes that is included while the libraries is prepared, the double-stranded DNA or the RNA will be unwind, and then one strand will pass through the nanopores. And then because the DNA molecules or RNA molecules, they are kind of carrying charges. So while the strands are passing through the pores, the electrical signals is measured while the sequencing is ongoing. So this is basically how the direct sequencing of RNA or DNA molecule is carried out. So from here we captures the base orders or the sequence, and then we also captures any epigenetics informations like the methylated bases throughout the process. So the process do not involve PCR. Actually, that means we do not need to run PCR to enrich the libraries before we do the sequencing. So we can have the native DNA genetics and epigenetics information from the sequencing run. And then the sequencing is rate length agnostic. That means even though nanopore sequencing or ONT sequencing, they are categorized under the long rate sequencing, but it's a little bit misleading because not only it can sequence very, very long sequence up to MB in size, it can also sequence very, very short sequences, less than perhaps 100 base pair or less than 50 base pair. So it doesn't matter whether your samples is of very long molecules or they are of very short molecules. They kind of, nanopore is not really picky in terms of the molecule length. And then the real-time analysis, like mentioned, so it can measure the electrical signals while the DNA molecule is passing through the pores. And then when these signals are obtained, we can do the real-time base calling and also to get the data immediately while the sequencing is still ongoing. Because the signals is captured real time, that means we can also ask the software to control or to monitor the DNA molecule that is passing through the pores. So with the kind of development of adaptive sampling, so the adaptive sampling is we inform the software that what are our target molecules inside the samples. So the software will help to monitor whether the sequence or the molecules that is being passing through the pores is the one that we are looking at. And then if let's say it's not really sequencing the one that we are looking at, then it will kind of eject the DNA molecule out. So it will reverse the signals or the electrical kind of the signals to ask the pores to eject the DNA molecule or RNA molecule out from the pores and then sequence the next one. So in more kind of details, it looks like this. So inside the software that is used to control the sequencing, we can provide the information like what are our targets. And then during the sequencing process, the software will monitor the DNA molecule when it passes through the pores. And then if let's say it's not our targets, it will be ejecting the strands from the pores and then continue for the next, kind of waiting for the next strand to pass through. If let's say the molecule passing through is our targets, then the sequencing will continue to proceed to obtain the sequence data. So this is the adaptive sampling, which is very unique in nanopore sequencing. And then at the moment, I think it's not seen in other technology yet. So the sequencing device. So before we look into the sequencing machine, these are the kind of the flow cells. So this is where we put in our DNA samples, And then this is also where the signals are measured while the sequencing is running. So there are two types of sequencing flow cell. One is the mean ion flow cell. The throughput for this mean ion flow cell is lower. So for simple cases like sequencing a bacterial genome or sequencing perhaps the amplicon metagenomic samples, normally the mean ion flow cell will be sufficient to provide enough data throughput for research purpose. And then it basically can give roughly 10 to 20 GB. And then actually from the recent runs that we have seen, I think the data throughput now can exceed more than 20 GB if let's say the flow cell is performing well and then the samples is kind of clean and then the pores are healthy. I think there should be no problem to even achieve more than 20 GB for now. And then it's suitable for small genomes. And then, of course, if let's say you just want to have some low-pass sequencing of large genome, like detecting the copy number variations or big structural rearrangement in the genome, it's still okay to use the mean ion. But for human genome or human whole genome sequencing, or the one with the adaptive sampling, even though the adaptive sampling is a targeted sequencing, most of the time we still need a larger throughput, data throughput. So that's why the other flow cell is there. This is called the ion flow cell. It can generate, I think, 100 to 200 GB of data per flow cell in one sequencing run. So it's kind of, the data throughput is just nice for one whole genome sequencing. And then, of course, for other larger genomes, this one will be also suitable. And then depending on the flow cell that is used for sequencing, there are different choices of sequencing devices or sequencing machine. So for mean ion flow cell, at the moment, there are two different machines available to kind of work with the mean ion flow cell. So one is the mean ion MK1D. This model is very small, so it's less than the size of my palm. And then the other one is grid ion. This is a larger scale or higher throughput of sequencing machine. So the mean ion MK1D can cater one mean ion flow cell at one sequencing run. And the grid ion machine can cater up to 5 flow cells in one sequencing run. So it's kind of, if let's say you are having a lot of samples, and then you need to sequence many flow cells at one run, then the grid ion could be your choice. And then the grid ion also comes with the compute on board, so you do not really need to kind of prepare another computer in order to do the sequencing. And then for the ion flow cell, at the moment, I think we can focus on two. So one is the ion 2 integrated and the ion 24. So both these machines come with the compute. So that means because the process of the base calling, that converting the electrical signals into the bases, the process will need the GPU power to do so. So that's why these two machines come with the GPU, and then the base calling can be done swiftly using the two machines. If let's say you are having a ion 2 solo, so this machine do not really have a GPU card inside the machine. In that case, the user will need to prepare a computer that is equipped with a GPU, a relatively good GPU card to do the sequencing. So perhaps a gaming laptop or gaming computer will be suitable to do the base calling for the P2 solo over here. So from the name, you can also see the numbers. So P2 solo and also P2 integrated, both these devices can cater 2 ion flow cell sequencing run at one go. And then the ion 24 can kind of do the sequencing for up to 24 flow cells at one sequencing run. So into the workflows on how we normally perform the tumor normal sequencing. So before we look into the details of the workflow, just want to clarify some of the terms that we might be using today. So one will be the somatic variants. So this is actually the title for today's webinar. And then how is it compared to the germline genomic variants? So for genome sequencing or DNA sequencing, most of the time we want to know what are the variants. So some might be using mutations instead of variants to tell the difference between the DNA molecule or DNA bases in the samples compared to the reference. So there are two types of genomic variants. One is germline, another one is somatic. Germline mutations or germline variants are basically the variants that present since from the gametes. So when the gametes develop into fetus and develop into the whole organisms, the organism or the cells supposedly to carry the variations or carry the variants in all the kind of across the whole body. So these variants will be passing through to the offspring, to the next generation. So these types of variants is considered germline variants. So in other words, they are also the inherited variants, and then these kind of variants normally will be passed through to the next generation as well. And then another type is somatic variants. So this one is kind of originally is not there in the body, but throughout the development process, some of the cells might start to have some variation or mutations occurring. And then these variations normally will be at certain parts of our tissue or at certain parts of the body. So this normally will not be passing to the next generation because they are only present in part of our tissue. And then unless the somatic variants happens in the germ cells, or else normally it will just stay to the kind of the individuals, and then it will not be kind of shared to other individuals by kind of through the germ cells. So how do these actually related to because when we look at somatic variations, most of the times one of the commonly studied cases will be cancers. So why somatic variants are studied in cancers? So cancer, there are also, I think generally we can put into two categories. So one is the inherited cancers. So the parents might carry some mutations and then pass to the next generation or offspring, and then the offspring also carried some mutations that will end up with cancers. So that one, if let's say we are looking in inherited cancers, most of the time we are looking at the germline variants. But for cases, I think most of the cancer cases is categorized under the somatic cancer. So that means it happens while it's not passing from the parents, but it's kind of developed while the individual is maybe exposed to some risk. Yeah, so this is some kind of the pictures on how the somatic mutations can happen in the tissue, and then how it's going to look like in the tissue. So throughout the process, like we have the normal cell in our body, and then maybe one day because of some exposure to certain risks, and then some part of it is damaged, and then the damage is not correctly being restored, then it will kind of end up with mutations that can cause some bad events. So this one, perhaps it will kind of make the cells kind of reproduce uncontrollably and then end up with tumors. So this kind of tumor cells, it will keep on growing without kind of the body would have no mechanisms to stop it accordingly. So it will become the tumors, and also if let's say in a worst scenario, it becomes the metastatic cancer cells, it will spread, kind of keep on reproducing and then spread across the different parts of the tissue and across different organs and even across different systems. So when we study the cancers, we would like to know what is the mutations that is carried in the tumor cells compared to the normal cells. So that's why the somatic mutations or somatic variations are normally the focus of cancer cases. So for identifying the somatic variations in cancer samples, actually from the DNA sequencing data, we are seeing the differences between the samples and the reference. And how do we know some of them are germline variations or whether they are of somatic variation? So the very common practice to identify whether the variance is germline or whether they are somatic, normally the experiment will be designed in a way from the same patients, we will be obtaining two different tissues or two different samples. one will be the tumor samples, another one will be the so-called normal samples. So if let's say a patient is identified with carrying a cancer tumors in perhaps a liver, then we will in order to identify the somatic variants that is inside the liver tumors, the samples will be collected from the tumor samples inside the livers. And then for the normal sample tissue, ideally the tissue will be collected from just beside the tumor samples. So let's say we have the liver organ, and then where we have tumors inside, kind of in one location, and then the tumor samples we are obtaining, kind of getting the biopsies of the tumor from the tissue, from the liver, and then the normal sample could be other non-mutated tissues inside the same liver organ. So we have the kind of, this is called the pair tumor normal samples. And then this one can help us identify what are the variants that is carried in the liver tissues, and then what are the mutations that are carried by the tumor tissues. And then when we have these two samples together, we can kind of minus off the variants that is happening or that is observed in both the tissues. Normally that one will be the germline variations. And then if let's say there are variations that only can be seen in the tumor samples and then not found in the normal samples, then those variants collectively be called as the somatic variants. So this is the normal way of identifying the somatic variants inside tumor samples by comparing to the normal samples. So of course, for other whole genome sequencing, if let's say it kind of we are studying some inherited disease, or we just want to check their variations, whether some of the mutations is observed in certain patients, but normally kind of the one sample whole genome sequencing might be carried out. And then for tumor samples or cancer patients, some cases we might also obtain only the tumor samples to do the whole genome sequencing, and then we compare to populations data so that we can know which one are the common variants that is found in the population, and then what are the variants that are only found in the tumor samples. But this one is kind of less resolved compared to the paired tumor samples, where we directly minus off, or we directly know what are the normal variants that is carried by the patients, and then compared to the tumor samples, what are the specific variants that is only found in the tumors. So for ONT, they have developed the end-to-end workflows on how to perform the pair tumor normal sample sequencing by using the ONT devices. So with the ONT sequencing, basically when we supply the native DNA to the sequencing process, we are able to capture the single nucleotide variants, structural variants, copy number variants, short tandem repeats, and also the epigenetic modifications. So it's kind of one set of data, one set of sequencing run. We are measuring multiple omics informations for the same samples. So normally we will be using the ligation sequencing kit to do so. So in more details, is shown over here in this picture. So from the patients, when the normal cell and also tumor cells are collected, each of them will be prepped into one samples or one library. And then for one libraries, we will be using, we will be doing the sequencing run. So one thing to note is that because normal human genome sequencing, we're normally looking at 30x sequencing coverage. That means human genome, we have 3 GB of data. And then in order to get sufficient data to do the analysis, we need to have 30x of the 3 GB. That means we need 90 GB of data. But for tumor samples, because it's complex, the recommendations will be, normally we will need to have at least 50x to 60x coverage for the tumor samples. That would also means we would need around 180 GB of data per tumor samples. So if we are having a pair tumor normal samples, we might need to use up three primary ion flow cells, assuming one primary ion flow cell is giving us 100 GB of data. And then in order to accurately identify the variants inside the samples, and then the recommendations for the base calling will be using the sub or the super accurate mode. And then for the kind of, if let's say you want to kind of have a quick look on how tumor variants compare to the normal variants, so this is basically the pictures of it. So when we have normal samples and tumor samples, and then the samples have been sequenced and then mapped to the reference. So for the germline mutation, we will be seeing the mutations across the normal samples and together in the tumor samples. But for somatic mutations in the tumors, normally the variants will be detected in the tumor samples, but not found in the normal samples. So this is how we differentiate the somatic variations from the normal germline variations. So sequencing, that's kind of from the sequencing or from the samples all the way, we have two samples, pair samples, and then when this pair sample put into one analysis, we actually have the analysis workflow or pipelines on running the analysis part. So we do not really need to manually compare the normal samples with the tumor samples in order to identify the somatic variation. Sorry. Okay. So for data analysis, I mentioned we do not really need to manually look into each of the variants and then compare the tumor and normal to see whether they are somatic. So there are already analysis solutions prepared and designed. So for ONT, basically they are data analysis or data handling solutions for all the users. So here we are looking at two different software. So one is called the Minno software, another one is called the EPI2ME software. So these two software are provided directly from ONT. And the Minno software is basically the device control software. So this is normally, we use it to control or monitor the sequencing runs. And the EPI2ME software basically is the one that will help us do the data analysis. So this EPI2ME come with the pre-configured workflows shared by ONT. And then these workflows together with the software are fully supported by ONT. So because the ONT sequencing or the nanopore sequencing have been used by many users and then adopted in many different domains, so that's why there are also a lot of public software available out there for ONT users to manage or analyze the data. So but most of the software developed by the public or the community will be in the command lines. The one that developed by ONT, the Minno and EPI2ME, both of them come with the graphical user interface where users do not really need to learn how to do commands in order to do the analysis. Other software from especially the one from the communities, most of them will be in the command line interface. And then if let's say command line interface is still a preferred way to do the data analysis, the EPI2ME workflow can also be run by using the command line. So inside this Minno and EPI2ME, the modules include kind of the modules included in the software will be the base calling, the QC, the analysis, all the way to report generation. So it will help to summarize the analysis that have been carried out without user kind of need to dig into every files in order to summarize the outcome from the analysis. So just touch a little bit on the base calling. So as mentioned just now, we have different options in order to do the base calling. So basically we have three options or three choices. One is fast, another one is this high accuracy base calling, and then the ones recommended for human or tumor normal sample sequencing will be the sub, which is the super accurate mode. So these basically are the three different modes for base calling. And then during the sequencing run, normally we need to enable the modified bases to be discovered at the same time by using either the high accuracy base calling mode or the super accurate base calling mode. So normally in human or in animals, we are looking at the 5MC and the 5HMC, perhaps in the CG context or in all types of contexts. So this one, the settings need to be set in the sequencing software while the sequencing is carried out or during the base calling process. So just to further look into what is the 5MC and 5HMC, 5MC is basically the methylated C. So we have cytosines in the DNA molecules, and we normally this molecule can be methylated in the natural form in the cells. So when certain genes is covered by the methyl groups of this kind of the C's inside the gene is all covered by the methyl groups, then it's like there's a protective layer for the DNA molecule. And then normally these methylated genes will not be expressed in certain conditions. Of course, this methylation is also something flexible. It won't affect the sequence of the genes, but it can control the regulation of the genes by either cover the whole gene with the methyl groups so that they are not expressed, or they release the methyl groups of the genes so that they are expressed in certain conditions. And then the 5HMC is another type of methylations. So instead of the methyl groups, actually it's hydroxyl methyl groups that is attached to the genes. So normally human or mammals, if plants, we are looking at the 5MC and 5HMC. And then for bacteria, the different types of methylations can happen. Normally will be the 4MC and 5MC. And then there are also methylations that can happen in the adenine, 6MA. So it depends on your kind of objective. So which types of methylations or epigenetic information that you are looking at. Most of the time we are looking at 5MC and 5HMC. And then this basically kind of can be directly observed in the sequencing data after its base call. So different base calling models or versions, they will be able to provide different accuracies in terms of the methylations or methylation information gathered from the data. And then if let's say you are running transcriptome using the native RNA, and then you would like to also know the epigenetics of the RNAs, even more epigenetics information can be obtained. So like all these. So this one is just to touch base a bit before we go into the EPI2ME data analysis. So this EPI2ME program is the one that we are going to talk more today. It's actually a very intuitive or user-friendly software. So it comes with graphical interface. To use it, normally we just need to do some point and clicks, select the pipelines that we want to use, and then put in the data that we have, and then just run, and then we will be getting the results and the reports. And then the software is actually can be installed in different types of operating system, including Windows, Linux, and also Mac OS. So the workflows or the pipelines that has been packaged together with the EPI2ME, there are quite a lot. And then for human genetics, there are pipelines designed for trio samples. That means in some study or some projects, we are studying the parents together with the probams to identify whether there's any kind of what are the mutations or variations that is carried or inherited from the parents. And then, and then normally for normal sequencing, if you want to perform haplotyping or phasing, that means from one single individual, we want to know whether the variants are, or the alleles are coming from one parent, or they are actually carried carried from different parents. So normally we will need to do some phasing. Normal ways of doing the phasing is we sequence the parents and the offspring. Then we can tell which variants is inherited from the father and which variants inherited from the mother. But for the long reads, because we are able to kind of identify the variants, whether they are on the same DNA molecule, that means we can perform physical phasing. So we can perform the haplotyping or separate out different alleles based on the theoretical parental information. We do not really need to have kind of the parents' data in order to do the whole genome phasing. So with long reads, if let's say for samples that we want to identify the haplotypes, we can directly do the kind of choose to use the physical phasing. And then other pipelines include the human variation. So this one, if let's say we are just looking at the normal germline variations or just identifying whatever variations inside a sample, then we can be using these human variation workflow. And then, of course, for tumor normal sample sequencing, because the analysis is more complex, we need to compare the tumor samples against the normal samples. So that's why an additional, more design pipelines is there. This is the somatic variation pipelines that we are going to look into it later. So other than that, it also have the workflows to analyze the metagenomics data, and then the workflows to study the transcriptome, and then workflows for bacterial genomes and etc. So, and then this one also to highlight here, because human genomic or whole genome sequencing of big genomes, normally the file size, the data throughput is quite high. So that's why the CPU requirements is normally larger compared to other pipelines. And then another thing to note is ONT actually provide free cloud resources for the users to run the analysis. But this cloud resources is not available for human-related analysis because human data or human genome sequencing data is kind of sensitive. So that's why even though ONT do provide the free cloud compute, but we are not able to use it to analyze our human data. So for human genetics or human genome data analysis using EPI2ME, we need a relatively powerful computer in order to complete the analysis. So for cancer tumor normal comparison in single workflow, so the analysis include the quality stats, identify the SNPs, the sharp indels, insertion deletions, and then they are using the cancer specific or somatic specific kind of software in order to do the analysis. So the software set or the software or the programs that is used in the somatic variation pipelines will be different from the normal human variations pipeline or human genome analysis pipeline. The software will be slightly different, but all this will be kind of end up with reports, and then we can visualize the results in perhaps genome browser in order to monitor closely or investigate further on the data. So the workflow normally is start from after getting the samples, and then prep the samples into the libraries, and then look into the sequencing device. And then when the sequencing is carried out or after the sequencing is carried out, we gather basically the files that are coming out from the sequencing. So this data from the sequencing run will be the input for the data analysis pipelines. So the analysis pipeline will be using the somatic variations. So the analysis, like I mentioned, align the data against a reference genome we set in the software, and then the pipelines will move forward to identify somatic short variants, including the SNPs and also the insertions and deletions. And then it will also help to identify any somatic structural variants. That is basically the structural variants that is only found in the tumor samples. And then we can also identify modified bases inside the tumor samples compared to the normal sample as well. So the output include the HTML reports, the alignment found in CRAM, and then the modified base information in the MAC format, and other variants inside the VCF format. So these files, all this analysis is considered a secondary analysis. And then if let's say we want to identify even get more insights from the data, we might need some tertiary analysis to look into in more details each of the SNPs or each of the variations. So from here, before I start the EPI2ME app demonstration, just want to walk you through some of the details for the demonstrations, because during the demonstration I might not be able to kind of show you kind of visually on some of the information. So for the compute requirements, in order to do the somatic variation analysis, the recommended computer specs will be having 64 CPUs, and then the RAM or the memory is ideally to have more than 256 GB of RAM. But this is the recommended requirements. Normally, if the computer passed the minimum requirements, it should be okay to run the analysis as well. Just it might take slightly longer time if, let's say, there are a lot of samples to be analyzed. So for the approximate runtime, so it depends on the sequencing modality. So whether we are looking at the whole genome or looking at certain regions of the certain regions of the genome, the running time will be depending on this. So a complete analysis of 60x tumor and then 30x normal samples with default settings will take around 6 hours plus. So yeah, it's actually taking some time to do the analysis. And then another note is that ARM processor support is forced. That would also mean if, let's say, we are having the Mac computer like the Mac Studio, even though it's very powerful, but because the processor, the Apple Silicon, it's kind of, they switch from, after they switch their chip from the Intel chips to the M1 all the way to M5, all of them are considered under the ARM processor structures, and the Mac computer will not be able to run the pipelines that is inside this EPI2ME, even though there should be no problem to install the EPI2ME, and then should be no problem to use some of other workflows. But this particular human variation pipelines, likely the pipelines will fail if, let's say, we are using the Mac computer. And then to do the analysis, of course, we need the input data. So the input data that is required first will be the reference genome in the FASTA format. And then we also need the data, sequencing data. So single tumor samples either in one BAM file that is the output directly from the sequencing run, or a folder of BAM files that is directly from the sequencing run. So this one, once the sequencing is done, normally the output we can choose to be in FASTQ or in the BAM. But if, let's say, modified base is something that we are looking at, the data will definitely be in the BAM format. And then if, let's say, we forgot to tick or forgot to kind of output the BAM files, and then we do not really identify modified bases during the sequencing run, then we might need to rerun the base calling and then converting the raw signal files of port 5 into the BAM files in order to do the analysis. And then this pipeline have now been updated. Now it actually can do the analysis if, let's say, a user is only having the tumor sample sequencing. So the normal samples data is now optional. If, let's say, no normal samples is provided for the sequencing or for the analysis, the pipelines will be using some panels of normals. So that means they gather some information from public database on what are the normal variations that are commonly identified, and then compared to the tumor samples that we have in the run. So it can help to take out whatever normal variations based on the panels of normals, and then whatever remains will be considered as somatic variations. So the folders can be arranged in two types. So we can have one single BAM file for one sample. We can also have multiple BAM files for one sample stored in one folder. So for tumor-only mode, basically need to take care of certain things. So now after the updates, we are able to identify the modified base, the SNPs, and also the structural variants. Basically, it's almost the same compared to the pair tumor normal analysis pipeline. Just that the software used might be slightly different. And then user need to take note of certain several things, because we do not really have normal samples to kind of minus out whatever germline variations that is identified in the tumor samples. So that's why some of the concerns will be, if, let's say, we are using the panel of normals that is having some differences compared to our samples, some of the germline variants will be missed out. So especially the one that presents in the perhaps in our country, but the panels of normal use is kind of the global panels. Then some of the results might be false positive, might be identified falsely as somatic variations. So these are some of the considerations or some of the things that the user might need to take note while running the tumor only mode. And then the demonstration, I'll be using the very, very small data sets. Actually, it's only partial chromosome 20 kind of data. So that's why the analysis that I'm going to demonstrate to you is going to be quite fast. Okay, so this is how the EPI2ME programs looks like if once it's started in our computer. In order to get the EPI2ME program, this is where we can download the software and install in our computer. So just we just need to go to epi2me.nanoporetech.com is a subdomain from Nanopore Tech website. So over here, just need to scroll slightly down and then click on the download button. And then here are all the installers for the latest EPI2ME software. And then depends on the operating system that we have for our computer, we can choose the installer accordingly. And then how to install, there's a very complete installation guide over here, showing how to do the installation on Windows and then for the Mac OS and also for the Linux computer. So this installation guide is very detailed, even all the screenshots of what you might encounter, like the prompts, is also included over here. So the installation process is actually quite easy and quite fast. It's just like the normal installation process in Windows. You just need to double click the installer and then next all the way and then follow through the process. Then it should be no problem to be kind of ready to be in the computer. So once it's installed and when we open, normally the first page that will be shown to you is a login page. So you can sign up a free account using the login page. And then, of course, you can also choose to continue as a guest to use the software. The benefits of logging in as a member will be we are able to use the cloud resources provided by ONT free. And then for users who continue as guests in the software, we will not be able to run any analysis using the cloud. So over here, once we enter this home page, we are actually seeing a dashboard where the kind of the status of the software is shown over here, and then some activities. And then if let's say there's any news from ONT, they will also be sharing the information over here. And then if let's say a later version of this EPI2ME software is available, they will also give some notification over here and ask you whether you want to update it. If yes, there's a button there for you to just click, and then the software will help you to get the latest version and then update the software in your computer. And then this is the first page. And normally for the first time, I will be looking into the settings as well on the lower left corners over here of the software. So this is to check whether the software has been set up accordingly. So normally we can take a look at the working directory, whether it's okay for the EPI2ME software to do the analysis over here in this location. Of course, it needs to have the permissions to read and write inside these particular folders and then to do the analysis. Other than that, I will be also looking into the locker tab just to make sure the system is set up accordingly. So it needs two programs to work. One is the WSL, another one is Nextflow. If this one have not been set up accordingly, we can also do the setup over here by open the setup and then do the setup over here. So this actually can help the users. User do not really need to go to the WSL or Nextflow website to identify what are the things that need to be installed in addition to the EPI2ME. So EPI2ME will help the users to identify the right one and then do the installation accordingly. Over here at the bottom here, normally this is also the place that I will also do some modification. So this one, it's actually how much computer resources that we want to allocate for EPI2ME to do the analysis. So at the moment, maximum it can use around 28 cores in my computer, and then it can use around 116 GB of RAM in my computer. So we can adjust this accordingly on how much we want to allocate for the software. Normally, I won't be use up all the computer resources for EPI2ME, because the normal Windows operations will also need some CPU and RAM. So I will try to kind of buffer a bit, or kind of allocate, kind of leaves a bit of space for the normal Windows operations to do its work. So I won't be using up all the computer resources over here. So once we have decided, then we can do the save settings over here. So once it is saved, then it will try to kind of inform the WSL or the dependencies over here, so that they are aware of what is the maximum that can be used. And then when running the analysis, then the EPI2ME software will try to allocate the resources according to what we have for the EPI2ME. So let's say some process is using perhaps 8 CPU, and then because we are allocating 28 CPU over here, then we can actually run 2 processes at the same time, or even 3 processes at the same time. And then when we submit the 4th process, the next flow basically will help to arrange the resources, because we might not have sufficient 8 cores or 8 CPU for the 4th process. Then it will put the 4th process into a queue, so waiting for the other process to finish first. So it's kind of, it helps with managing the process, managing all the tasks, so it won't crash the computer. So once the setup is okay over here, when we are happy with it, then the rest, actually, I think at the moment we can skip all of them first. It's not that important for users to do the analysis. Most of the time, we are just using 2 buttons over here. One is the launch button, another one is the analysis button. Launch button is where we choose the pipelines that we want to use for analysis. And the analysis button is basically where we see the results from that for the analysis. So for somatic variation pipelines, where is my pipelines? Yeah, this one. So you can see over here, the computer icon is light up. That means I have installed the pipelines in this EPI2ME. And then the cloud icon, even though it's light up, but supposedly if we are looking at human samples, we shouldn't be using the cloud. So one clicks into these pipelines, the full documentation or the details of the pipelines will be shown over here. And yeah, that include the compute requirements, and then the input and also the output from the workflows. And then the pipelines overview will show step by step what will be carried out in the pipelines when we do the analysis. So including like the parameters and also the software and versions that will be used. And then sometimes it will also give some notes on what we should expect from these pipelines. So troubleshooting guide and FAQ also available here. And then here, when we want to do analysis, we just need to click the launch. And then before the launch, actually some workflow management options are available over here. So this one, I'm going to skip it first, and then just click the launch. So for the somatic variations, here we have option to choose what kind of variations that we want to identify from our data or our samples. So this one can be multiple choices. So if let's say I want to get all the short variants, the structural variants, and also the modified or differential modifications for my samples, I can kind of choose to tick all. And then under the main options, here we can put a name for our samples if let's say we want. So perhaps I put it as demo. And then for the normal BAM, so this is the data, sequencing data for the normal samples, and then this is the sequencing data for the tumor samples. So we need to provide, the normal sample is optional. So actually I have run another analysis using the normal and tumor samples. So I'm going to try out, let's say I only have the tumor samples. So in this case, let me choose the tumor samples BAM file. So once click into it, depends on whether we have one single file or a folder of BAM files. So let's say if our files is in single BAM file, we can choose the files over here. And then in the file explorer, we browse or navigate to choose the BAM files that we are going to use as input. Once chosen, it will be shown over here. So normal sample, I'm going to assume I do not really have normal samples over here. I only have tumor samples. And then reference genomes. Supposedly, we should be using either the HG19 or the HG38. So it depends on your preference. The recommended one is the reference genomes with no alternate contigs. And then this is actually only the chromosome 20. So I'm going to speed up the process. So this is a small subset. For the whole genome sequencing, supposedly we need to put in the whole genome of either HG19 or HG38. So with this, actually, you can see all the options are already in the status of green ticks. That means actually this analysis is ready to be carried out. But I'm going to change some of the settings to fit these data sets. So I'm not going to put a target regions for the data. So I'm going to ask the pipelines to analyze everything that I put in, or basically the whole chromosome 20. And then if let's say we want the software to skip some repeats regions, we can also supply the pipelines with the where the locations are those repeats, and then it will try to skip those complex regions. And then here we can also provide a panels of normals. So especially like cases what I have right now, I only have tumor samples. Ideally, we should supply a panel of normals so that it can kind of help to identify the somatic variants or separate out the germlines from the somatic variants. And then here, by default, it will help to do the SNP-F annotations, and also will be also provide the output files for IGV visualizer. So I'm okay with this. The next one, quality control. Because I'm using a very small data set, so I'm going to set it to 0. So this one, it will try to only analyze the bits when it achieves certain coverage. So if let's say our data throughput is very low, we might need to set it to all the way to 0 or 1. And then because supposedly tumor sample, we should be sequencing or getting 60x coverage. So this one we can also adjust accordingly. And then because I do not supply normal samples, I'm going to leave it as it is. And next, small variant calling options. So here are some of the options or settings that we can adjust according to our samples. Here, I think I have nothing to adjust. I'm going to use the default. And structural variance calling, and then the minimum size it will try to capture is 50 base pair. Of course, we can also increase it or decrease it accordingly. But if let's say it's too small, the data will basically be captured in the short variance pipelines. And then for the methylation calling, we have options to ask the process to be kind of strand specific. So we want to perhaps detect the modifications separating the forward strand and the reverse strand. So in that case, we can tick to force the strand over here, and then to perform the differential modified basis comparing the tumor and the normal samples, if let's say we have the normal samples. And then the multi-processing, as mentioned, each of the pipelines, it comes with the settings on how much computer resources will be used for the pipelines. If we want the process to be faster and then we do have sufficient computer resources, we can choose to increase the number of CPU or threads that is used in the pipelines with the hope that it will shorten the analysis time. But by default, normally I'll be using the default settings over here. Unless I'm in urgent, I want to kind of speed up, then I will maybe double up the numbers over here. And then under the advanced options, this one, based on the data that we have, actually, they have taken out the base caller information from the BAM file. So that's why in EPI2ME here, I need to specify what is the base caller that is used to generate the data. So for your case, if let's say the data coming out from the sequencing machine, normally we do not really need to change the settings over here. This is because the demo data, they have taken out the information, so I need to specify it so that the pipelines is aware of what kind of base call model that is used to generate the data. And then other than that, this one is just sending some ping to ONT, so they just want to check, kind of keep track on how frequently a pipeline is used. So this one is okay to tick or untick. It won't affect the analysis. And then lastly, under the next flow configuration, we can choose to give a name to the analysis run. So if let's say we do not really put in anything, by default, it will be using a random name for the analysis run. Because the name is too random, so sometimes it's a bit hard to tell what are the analysis that we have carried out for each run. So normally I will try to give some more meaningful name so that when I look at the name, I know what I'm doing for this run. So this one is a demo, and then it's run on 25th June, and then tumor only. So yeah. So once I'm happy with all the settings, just click the launch workflow to start the analysis. So the EPI2ME will check out whether it has all the information that it needs to run the analysis. And then once the analysis or once the check has been done, then the analysis will be kind of initiated immediately. So when we are seeing this page, that means the analysis is now has submitted to the computer and then is in the status of running. So once it started, we can see some progress over here. But because even though it's a very, very small data set, it still will take, I think, at least few minutes. So I'm going to show you the one that I have completed yesterday. So this is actually a run by that where I put in the normal samples and the tumor samples, and also using the same chromosome 20 as my reference. And then this one, I also have put in the demo data or demo targeted regions for doing the analysis. So for this one, as you can see over here under the details, the tasks that have been carried out in the analysis, quite a lot, many steps. And if we are seeing all the green bars over here, that means all the tasks have been carried out successfully. And then if let's say the status is completed in green color, normally that also means the whole analysis have been carried out successfully without any errors. So once the analysis is done, we can click on the reports over here to look at the result summary. So under the reports, normally for a complex pipeline like this, we will have more than one report. So here we are having a report for overall. We are also having a report for the sequencing data quality, and then individual reports for different types of variations. So we have one report for the SNPs or sharp indels, and then we have another report for the structural variants. We also have the reports for the somatic modifications information. So let me start from the overall reports. So this one, it actually kind of gives us the status for each of the sub workflows that we are looking at. So all the QC and SNPs and structural variants and the modifications informations, all the analysis have been carried out successfully or passed. So and then we can proceed to the next report, read QC report. So this one will summarize for us how much data we have submitted for the analysis. So in this case, we have only 13,000 sequences for the tumor samples. We only have 7,000 plus for the normal samples. And then what is the read length for the tumor samples, and then the read length for the normal samples. And then for the coverage is roughly 3.4x, and the normal sample is only 1.4x. So it's a very, very small data set, and it's not even sufficient to cover a very small region in the chromosome 20. So from here, it also tells kind of the details on the sequencing data, like the read length distribution, the quality, and the quality as well. And then how many of them is mappable to the reference, and then how many of them is not mappable to our reference genome file. And then the coverage across the reference. So from here, we can see actually across the chromosome 20 is only covering a very small kind of multiple regions inside chromosome 20. And then same for the another normal samples. So when this looks okay, then we can proceed to look at the variations that have been discovered from the samples. So perhaps we start from the SNPs. So here are all the somatic variants identified from the samples after they compare the tumor samples against the normal. So the total 45 variants, including 38 SNPs and 7 indels. The transition traversion ratio is also stated over here. And then inside the reports, we also have kind of the details of the small variations identified. Yeah, so this one I'm not going to go very details. Just want to show actually the CleanVar annotation is here. Even though CleanVar has not been popular, kind of not really used for somatic variations annotation purpose, I think they have just started to keep the somatic variation information since I think 1 or 2 years ago. So the informations for somatic variations from CleanVar might not be a lot. So that's why we need to proceed to kind of annotate with more robust database if we want. And then at the moment, it's not those more robust cancer-related database is still not covered in EPI2ME pipeline yet. So we need to manually do it after we obtain the SNP data from here. So other than SNPs, we also have the report for structural variants. So here we have actually zero number of somatic insertion and somatic deletions. Somehow we have other two structural variants. So this one is BND. From what I understand, is a breakpoint. So it's a translocation, somatic translocation identified in tumor samples, but not in normal samples. So some of the information are shared in the reports over here because our data set is too small. So at the moment, it's supposedly to be not a lot of significant or meaningful informations over here. But if let's say you have whole genome sequencing data, it's going to be a lot of informations over here. And next one will be the modified base reports. So this one, again, it will try to see the modified rate for the tumor samples and the modified rate for the normal samples, and then how many of them is differentially methylated loci and how many of them is considered differentially methylated regions. So it will try to compare, kind of gather the summary, like total how many Cs are identified in the normal samples, how many Cs identified in the tumor samples, and how many of them is unmethylated, how many of them is having a methylated, kind of the methylated Cs, how many of them are considered the hydroxyl methylated C. So details, all the output files are available and summarized in these reports, including the differentially modified regions. So these are the kind of the regions that is highly methylated in the tumor samples, but in normal samples, the observation is not there. So it's something interesting if let's say we want to see what can be the as kind of the modified base association with the cancer, then I think this could be some positive signals that these regions could contribute to the cancer profiles. So details, or not really a lot of details over here. So if let's say we want to get the total files, output files, we can switch to the files tabs over here. So from here you can see we are going to get a lot of output files from one pipeline analysis. So they have labeled the output files according to the types of analysis that have been carried out. And also, if let's say we are looking at certain specific files, we can also look into the specific sample files over here. But my preferred way of looking into those files will be I can open it up using the File Explorer. And then all these are kind of accessible directly from the computer. And then the reports are also provided as part of the outputs. So if let's say we want to share the results with our collaborators, we can share the reports in HTML to them. They can open up the reports in web browser like Google Chrome or Safari. They do not need to install the EPI2ME in order to view the reports. And then a very light version of IGV is provided here in EPI2ME. So we can have a very quick looks at which region is having the variance. But for kind of more robust visualizations, normally we will also recommend users to install a standalone IGV, which is also free, can be obtained from internet, from Broad Institute. And then the full version of IGV, we can put in the alignment files and all the modified base signal files, variance files, everything in one browser, and then visualize multiple data sets, multi-omics data in one place. So these are basically the output from the pipelines. The last tab is a logs. So this one, normally user don't need to look into that. We only look into this part when we do analysis and it run into errors. So from here, we will try to see the error message, what could be the reasons that results in errors in our analysis, and then try to do some troubleshooting from here. So if let's say everything is running smoothly, and then this page will just help us to double check some additional informations that we put in for the analysis. Other than that, I think the log file, you can just ignore them. So yeah, the one that I have submitted just now has been completed. It takes around 4 minutes. So everything looks okay. Same, we also are going to have the reports for each of the sub workflows that have been carried out, even though we only put in one tumor samples without the normal samples. So the results or reports will be similar. It's just that when comparing to when you try to compare to the panels of normal, it will be using the default panels of normals. So if you prefer to have a better results, you could also design some panel of normals to be included in the analysis. Then it will compare your tumor samples against your selected panels of normals. Yeah. And then one last thing, in case when we run analysis and it starts with errors, something like this. So as mentioned, we can go to the logs over here and then try to guess what happens based on the error message. But sometimes the message is not so intuitive for most of the users. So one way that I normally do will be copying the whole error message to ChatGPT or other AI and ask the AI to help to suggest solutions for or rectify the problems. Other than that, we can also choose to report issue over here. And then this will actually help us to package the log files into a zip file where we can share to ONT to get help from them. So when email them, we need to attach the log files, and then we also need to describe the problems that we are having when running the analysis, so that the tech support personnel will be able to identify the problems and suggest solutions accordingly. So other than email to Nanopore Tech directly, you can also reach out to NextGen to me in case you need some help in terms of running the analysis and EPI2ME. So yeah, I will be happy to help if let's say you have any problems. So yeah, that is all I have for the demonstration. So back to here. The analysis, if let's say you want to follow through the whole demo analysis, these are the settings that can be set. And then the demo analysis can also be run very quickly from here under the workflow control or management. So when we run the demo analysis, it's actually it will download the demo data sets and then do the analysis over here in the computer. So yeah, so the analysis that was carried out is actually following these parameters. So feel free to try it out when you have the EPI2ME in your computer. So the output files, actually the details of the output files is also in the documentations. So if you kind of want to know exactly what are those output files, it's actually a very long list. You can see kind of check by their file names and what are those files over here in the EPI2ME as well. So with this, for today's, we have gone through briefly on the nanopore sequencing technologies and also look into the EPI2ME program. And specifically, we have go deeper into the somatic variations pipelines and also look into the results and reports. Yeah, so that will be the end of my presentation.
Yeah, thank you, Jashin, for the insightful and comprehensive presentation. So now we will move into the Q&A session. You can unmute yourself to ask the question or feel free to type in the chat box. Is there any question from the floor?
I think so far there's no questions.
Yeah, so I will conclude the session for today. Before I conclude, we would like to hear your thoughts. If you have a quick moment, please fill out the survey form as I pasted in the chat. and we will send the e-certificate once it is ready. Also, if you have any further questions after this next talk session, feel free to contact us at sales@nextgene.my. And thank you everyone for joining with us today, and we are looking forward to see you in our next event. Thank you very much and have a nice day. Bye-bye.
Bye.